anatomists and physiologists. This is part 4 of our digestive series. This is digestion and absorption physiology. Okay, I love this picture a lot because it shows all of the different places that each foodstuff gets digested and absorbed. We'll discuss all of these main groups and how digesting and absorbing work. But I want you to note there's some important groups here. But before we talk about each of these, and I'll zoom in on some of them, we need to remind ourselves about some important stuff. So I'm going to draw this off to the side. So these molecules that are all listed here, they're all big molecules. They're called polymers, rather, polymer. Polymers are big molecules. So polymers, they're big molecules, and I'm simplifying this here, but like if we had a bunch of simpler molecules, and of course it's not always a chain, but polymers are like big molecules consisting of little bits, and each of those little bits are called monomers. So little bits, making up the bigger thing, which is the polymer. Okay, so all of these guys that we see carbohydrates, we see proteins, fats, nucleic acids. Those are the big ones, the polymers. And then the goal of digestion is to get them down to monomers, a smaller bit that we can actually use and actually absorb and then use in the body. Okay. Okay, so back to the picture. So when we think about something like carbohydrates are like sugars. Like starch and sucrose, which is table sugar. Lactose is milk sugar. Okay, there's that sucrose I was talking about. Maltose, grain sugar. And then we try to break it down into its really simple forms, monomers. Things like glucose, fructose, galactose, all of those different types of ones. We can use those in the body. Proteins are chains of amino acid. Where it's like amino acid, amino acid, amino acid. But then that protein gets folded up into this really like complex shape. And so we want to break it up back into its simple form and maybe back even into its own individual amino acids. So it's the monomers, right? Well, so we want to use those because we can use them in the body. Okay, fats are also known as lipids. And the lipids that we tend to eat in our diet are triglycerides. Triglycerides are the polymers rather, and then we want to break them up into things like fatty acids. We can use those in the body for energy. Nucleic acids are things like DNA and RNA. So DNA, RNA. So if there's anything that we eat that has DNA or RNA in its cells, then there's enzymes in the pancreas that break them down into nucleotides. So those nucleotides are the monomers, and then we can break it down even more into those individual bits that make them usable in the body. What's not on this slide is the absorption of vitamins, of electrolytes, and water, but we'll go into each of those. Okay, so the next slides are just the zoomed in version. I want you to have the zoomed in version for when you're studying. This is also in your textbook, just so you know. This is figure 23.36, so just so you know. So these are just all the zoomed in versions of what I just showed you. Okay, so jumping in now to like how each of these larger molecules are digested. That's what we're jumping into now. So how are carbs digested? I'll start by talking a little bit about starch, but then we have some other ones to talk about too. So the first thing is that we have salivary amylase in the mouth. That's in the saliva. But remember that enzymes are proteins. Amylase is an enzyme, and enzyme's a protein. What happens in the stomach is the digestion of proteins. So once that amylase gets into those gastric juices, it goes away. It just doesn't work anymore and gets denatured. And so starch digestion halts. What amylase does though is it takes starch and makes it smaller. And then once we get to the stomach, we'll pause for a minute. And then the stomach does its thing. While we're in the stomach, we digest some proteins, do some other stuff. But the next time we see amylase is in the pancreas, pancreatic amylase. And all the stuff from the pancreas gets delivered to the small intestine, so that's great. This happens in combination with brush border enzymes. The brush border enzymes help take pancreatic amylase, so the starch, and then other monosaccharides or other sugars. We want to bring them into monosaccharides, which are the monomers of carbs. So the goal is, remember, that we want to take the bigger version of the carb, whatever that carb looks like, that's the polymer. We want to break it down into individual. We want to break it up into individual bits because then we can use those in the body, right? So here's a bunch of examples of enzymes that we can use. So for example, maltase breaks up maltose, that sugar maltose. Sucrase breaks up sucrose, table sugar. Lactase breaks up milk sugar, lactose. So folks who are lactose intolerant lack this lactase enzyme, which means we can't break up that lactose sugar. And that becomes a problem, right? Because if we can't digest lactose, the sugar, then the contents of our small intestine increases in its solutes, and then more solutes mean that we draw water. So that's osmosis. And that can lead to things like diarrhea, because there's more water, right? But the other thing is that we can start to digest it, but it doesn't happen till the large intestine, and that's when the gut microbiota gets to it. And that's a lot less elegant of a solution because that can lead to lots of bloat, lots of flatulence, and pain. So there's actually a really cool solution to this at this current time, which is taking a synthetic lactase enzyme, or like lactate, if you've heard of that before. If you're consuming milk products and you just so happen to be lactose intolerant, synthetic lactase. How cool is that? Anyways, but one of the molecules, once those molecules are broken up into these littler bits, the monomers, the monosaccharides, then we can take them into the cell. We take them into the enterocytes, the gut cells of the small intestine. We take them in from the apical portion because that's the part that's just like inside the tube. And it happens with secondary transport. Secondary transport means that we need a buddy to get into the cell. And that buddy that we need to get into the cell is sodium. Then we leave the cell on the other side, that's called the basolateral side, and then we enter the blood and then we get used. Used for energy. Or whatever we need to use it for. Let me show you what I mean. So this is carb digestion, and this is a picture from your book. And this is all in the small intestine. So the steps are numbered a little bit differently, but that's okay. So what happens is we have this molecule starch. Starch is a big old carb. The actions of pancreatic amylase break up that starch into smaller bits. Once it's small enough, like down into its constituent forms, then we can take that starch, that little bit of starch, that monosaccharide version of starch, with sodium, bring it into the cell. And all of these are like different enzymes of the brush border, that kind of thing. One of the words that your textbook brings up is oligosaccharides. Oligosaccharides are not quite polymers, but they're not quite monomers either. They're kind of like an in-between form, just so you know. Okay, so once those monomers, those monosaccharides are inside the cell, they'll travel across the cell, and then they'll leave the cell into the blood. So those monosaccharides, they're co-transported across that membrane. That's when they need their buddy, sodium. And they call it the absorptive epithelial cell, which is just the enterocytes. There's some parts about the sodium-potassium pump here. All this part means is that the sodium-potassium pump is helping all of this happen. That's all I care that you know about it this time. But anyway, so what happens is those monosaccharides will then go into the blood because they'll pass through that cell with that carrier protein that's facilitated diffusion there. Okay. So we break up that starch, break it up some more, co-transport with sodium, travel across the cell, leave, and enter the blood. That's what happens. That's carbs. Proteins happen in much a similar way, except the stomach is the first thing that we need to talk about first. Okay, so the stomach has this enzyme or this protein pepsinogen, which becomes pepsin, and that digests about 10 to 15% of our proteins. But pepsin only works in acidic environments. So when we get to the small intestine, it's really basic in there. So pepsin does not work in the small intestine. But we do have proteases from the pancreas. Prot refers to protein. And then that ase, that ASE, means enzyme. That's a common ending of an enzyme name. Okay. So the two main ones are trypsin, chymotrypsin. They digest small peptides, but we also have some other ones. Carboxypeptidases, they break up stuff into amino acids. And then we also have brush border enzymes. So lots of things are doing protein digestion. And they're all fall under the category of proteases, right? So we have trypsin, we have chymotrypsin, carboxypeptidase, and then all those enzymes of the brush border. There's some examples here. Aminopeptidases, carboxypeptidases. Anyways, you get the point. Oh, this is not supposed to be carb body. It's supposed to be carboxy. Carboxypeptidases. Anyways, so a similar thing happens that we take those amino acids, those monomers, we co-transport them with sodium into the cell, they travel across the cell and enter the blood. Let me show you what I mean. So this is starting in the small intestine, right? So what we have are little bits of protein. All of these balls are amino acids. Notice that we go from a little bit of protein, now we have individual amino acids, monomers. Those monomers can enter the cell with the co-transporter sodium, travel across the cell, which is in part motivated by the actions of the sodium-potassium pump, period. That's what I care that you know about. And then that amino acid is able to leave the cell, enter the blood. Lipids are digested a little bit differently because lipids are just larger molecules. Lipids are digested by lipases. Those are fat digesting enzymes. So here's how it works. It mostly happens in the small intestine. The first thing that we do is we want to emulsify those fats, and that happens with bile salts. Bile salts. because that fat will emulsify. And remember that emulsification means that we get like little teeny tiny beads of fat, which is lots and lots of surface area to act on, right? And then we get to digestion, which is pancreatic lipase. Pancreatic lipase. And remember that the type of fat that we eat is called a triglyceride. So we have this molecule that looks like this. It's a glycerol, which is a sugar. And then we have 3 fatty acids off of it. This is fatty acid 1, fatty acid 2, fatty acid 3. What pancreatic lipase does is it acts on that triglyceride. This is a triglyceride. Much, much simplified, okay? But what happens is we get 2 fatty acids, and then we get 1 monoglyceride. So it's glycerol with 1 fatty acid off of it. So do you see how that's already smaller? So we've broken off 2 fatty acids, and then we just have that 1 monoglycerol left. So we call that monoglycerol. The whole thing is that we're trying to make it smaller. Speaking of smaller, we start to form micelles. Micelles are extra tiny drops. Extra tiny drops of fat and bile salts. Like if you've ever heard of micellar water, it's little tiny drops, okay? And then the next thing is that once we have these extra tiny drops, then we have diffusion of those little tiny stuff into the enterocytes. This involves the micelles breaking apart, and this is because lipids are fat soluble, right? Because they are fats, right? Because all of that fat can pass through the plasma membrane. Those little bits of fat can pass through the plasma membrane, and so then we just diffuse into the cell. Once we're in the cell, we form chylomicrons. Chylomicrons. Chylomicrons are little bits of lipid with protein on them. So they're lipoprotein drops. So they're a lipid with a protein in the drop, in a form of a drop. And then we transport that chylomicron. Chylomicrons are too big for capillaries, so instead of entering the capillaries, we enter the lacteals. Remember, lacteals are specialized lymph vessels, right? And so the lacteals will then enter the lymph, and then from the lymph, we'll go back into the blood via the thoracic duct. Lymphatic system, coming back again. And then lastly, so we have still these chylomicrons, they're still pretty big though, right? So in the blood, we have yet another enzyme, and that enzyme is called lipoprotein lipase. So it's an enzyme that digests lipoproteins. So it breaks up chylomicron into fatty acids and glycerol. And all of those can be used for energy. And then the liver also plays a role in this too, a little bit. Takes over anything that's left over. So here's a summary of what we just discussed. So we have this little blob of fat, the bile salts emulsify, And then we digest with the pancreatic lipases. There's the emulsification step. Pancreatic lipases, and then we form micelles. So notice that those fat droplets get even smaller with the formation of micelles. Then that diffusion step occurs when we get fatty acids and monoglycerides. We get the formation of chylomicron, and then we transport it. And then that chylomicron ends up in the lacteals, and then we get back into the blood. And then those chylomicrons then are digested by the other enzymes, those lipoprotein lipases. Lipoprotein lipases digest chylomicron. But it's not shown on this slide just because the textbook didn't. Okay, the last little bit that we'll chat about here, and then I'll just show you some summary tables for your own edification, but absorbing vitamins, electrolytes, water, this is what we got here. This is what's left. So vitamins. With vitamins, the small intestine does a lot. The small intestine absorbs a lot of vitamins. The large intestine also absorbs a lot of vitamins, but it's mostly just the B vitamins and the K vitamins from its bacteria, from the microbiota. If vitamins are fat soluble, they get involved in those like fat micelles we were just talking about. So they just like get be little tiny and then we need to absorb them. Anyways, a lot of stuff. If those vitamins are water soluble, like B vitamins, C vitamins, there's some specific transports. For example, like if you're B12. So if you're B12, then intrinsic factor, which is released by some of our stomach cells. Intrinsic factor aids in uptake. Into cells by endocytosis. And that happens in the small intestine. Remember I told you the small intestine does a lot of that vitamin absorbing. Electrolytes mostly get absorbed in the small intestine. Electrolytes mostly get absorbed in the small intestine. So we have things like sodium and chloride and bicarb. Sodium and chloride mostly follow each other. And sodium oftentimes wants to go into the cell due to the sodium-potassium pump. Potassium oftentimes enters cells by diffusion. Iron usually involves active transport into cells, and it enters the blood when needed. And it usually hangs out where it can bind to this molecule ferritin, which kind of just holds on to it. And then once iron is needed in the blood, then it unbinds ferritin and enters the blood. Calcium is absorbed or not absorbed depending on those blood calcium levels. So we know that PTH plays a role, parathyroid hormone. Vitamin D, though, the active form of vitamin D does help with calcium absorption. Okay, and then lastly is water. And water gets absorbed a lot in the small intestine. Most absorption happens in the small intestine. The small intestine is because there's lots of water to absorb because it's osmosis. So then there's lots of water absorption. Lots of water absorption into in the small intestine. And then the large intestine does the rest. So large intestine does the rest. Okay, the last little bits of slides here that I'm showing you are just summary tables. I think all of these tables are really good to look at. And they're from your book. I think you should look at them. Nice little summaries.